you?
Hello. Hey, Sharonda. Hi, Dominique. Hello,
Hi, Dr.
everyone joining
Bailey.
us.
Hi,
How is
everyone.
how is everybody doing this evening? It is what, Thursday. Oh my goodness. The last day of November.
The last
The,
day.
the last day. First
Bye
of
bye,
all, where did
November.
the time go?
Not a clue. It's weird. Seriously, though, it's a little like I always feel like the year goes really fast, but this one has been a little uncomfortably fast for me.
Yeah.
It's
Exactly.
almost December.
And what are we going to do? Oh my gosh, it's almost time for 2024. And this is the last town hall. So I see people in the comments. Y'all already know me. You know
They
I like
already know. Listen, I love that you all are so ready because you already know. Tell us hello and where are you joining us from? Check that out.
they
They
already
already
know.
did it before you said anything, Sharonda.
They
They
know
used to it.
they're very used to it. So hey everybody, so this is the town hall. You are in the right place. Our town hall. I'm doing my Vanna White thing at the top. Yes. Welcome, welcome, welcome to the town hall, but welcome to the last day of November. Oh my goodness. Hello to my partners. I see y'all in the chat. Hey
I
everybody. Oh,
see you. Hello, hello.
hello. Oh, New York is in the house.
Hi, Dr. Marjorie. I'm in
She
New York.
said New
Know
York is
you.
where her heart is, but that's not where you are.
Oh.
Huh, Dr. Marjorie?
We
Yeah.
New Jersey is where my heart is.
We all know
It
that,
lives
Dominique.
in New
We
Jersey.
know that. We know you a Jersey girl. It's
North
a thing.
Carolina. Ooh, United Kingdom. Hey,
Oh,
hey, hi there in the
nice.
UK.
UK, hi.
We're
How
coming
are
to visit
you?
y'all next year. Hi,
Yes.
everyone. Hello,
Philly is
hello,
in the
hello.
house. Hey, Philly. Hey, Philly. Hey, Texas. Texas,
Got
how are y'all?
excited.
I love it. Thank y'all for joining us and letting us know where you are from. We appreciate it. We thank you for joining us for this town hall. But we just really wanted to know where you guys are from. Talk back to us. Let us know what's going on in your world. Yes, Dr.
And
Bruce.
let us know if you're a warrior or caregiver. We're all
Ooh.
family here. We're all sickle siblings. Let us know. We love connecting with all of you. So if you are up to it and willing, I am a warrior. I think the three of us here on the line are all warriors.
Oh,
Yes.
For this last
Warriors.
week, I've had the opportunity to also be a caregiver. So that has been an absolute role reversal. And y'all, I'm exhausted.
yeah.
So, caregivers, you guys are amazing. We just had a whole caregiver summit. And let me
Yes,
tell
we
you,
did.
ended the summit becoming a caregiver again for a full week at the bedside at the hospital. And let me tell you, I have a brand new appreciation. Crystal, I see you, warrior and caregiver.
Oh,
Oh, wow.
yes.
We have someone joining us from Belize. Did you guys see that?
I did not.
Ooh.
I
Yeah.
did not.
Hi
I want
there,
to go to
Los
Belize.
Angeles. Ismael. I love that. Ismael. Condo, Alabama, Boston.
Minneapolis, North Carolina.
Marjorie said, no, no, let's not get it twisted. I just live in New Jersey. I
I know
understand.
it. Dr. Marjorie, I'm the same way. No, no, don't get it twisted. I just live in New York, but I am a New Jersey girl.
She was like, let me make it clear. Oh, we have a researcher from the University of Cambridge. Ooh.
Oh,
Oh, and
on
they're
sickle
doing
cell
research
neuropathy.
with yes, we
Sickle
need to
cell.
connect with you
We need to connect immediately. Kevin,
immediately.
hi
Yes.
there,
Right?
Kevin.
Oh, yes, I already wrote down his name.
I should have known. We got Dominique on. She's definitely going to get those notes. We
Oh, yes,
appreciate you,
I
Dominique.
got them notes. Welcome and thank you for joining us. We got
Yes.
Joy. Welcome, everyone. Oh, we have a lot of warriors. We got a lot of caregivers. I hope everyone enjoyed the caregiver summit. I know I did. What about you,
Yes.
Sharonda?
You know I did. You know I enjoyed it. I like being on the background, so it was super fun for me to be trying to move the pieces in the background. But my favorite part, if I had to say, was um Mother Doris giving her speech at the beginning. And I'm gonna keep talking about it. I know people are getting tired. I don't care. I loved it. It was so good. I appreciated her session, and I absolutely loved the music, because me and my family, we all about music, so I love the music for the kids. So those are my two favorite. What about you?
Um, I think mine was I enjoyed the session, the opening, and I always say this. I'm not even gonna say the word because I always say the word wrong. But the opening session with Dr.
The
Azar.
plenary, yes.
Oh, when I
Yeah,
say
that was good.
absolutely
He
amazing, it
was
was absolutely amazing.
phenomenal.
Yeah.
It
Yeah, you're
it
right,
was,
Dr. Bailey. Phenomenal.
He
Yeah,
was phenomenal.
he really was. We already
and
wrote him in for convention.
Oh, already,
already.
already.
It's a
Kiara, your favorite was navigating the healthcare system with Dr. Bailey.
I heard she killed that session. I heard that session
Sarah,
was
you
really good.
already got the job, sis. You already got the
I can't believe you just said that.
job.
I agree, Carla. Dr. Azar was amazing. I think my favorite was, of course, my mother. Everybody said it. They kept saying in the chat, look how
Yeah.
proud Dr. Bailey is. And I am. Mama Doris is amazing, and I get to call her mine. I loved Dr. Azar. And then, of course, the um, being able to honor and award our caregiver heroes, our caregiver
Oh,
shout
yes.
outs. I love that so much. And I enjoy really, really just being able to acknowledge how amazing you all are.
Yeah. Well, I am super glad that we had that time at the Caregiver Summit. And yesterday we did our wrap up with the team, and we gave you guys a good recap and overview of, for those of you that couldn't make it. So just know, next year, please make sure that you signed up early so that you can be a part of it. But we have so many events in 2024 that's coming up. If you want to stay connected with the things that we're doing, make sure that you become a part of our newsletter, because that's where you're going to get most of the information. Partners, you guys already know. Just a quick note, tomorrow is our last partner meeting of the year. So make sure you are checking your emails. And just if you want to be a partner with the Sickle Cell Consortium, credentialing is now open, and you can become a partner and be a part of this great community. So make sure that you guys get that information as well. But we're about to get started and really talk to you guys about a cool company that has supported us throughout 2023, and we're so grateful for. And Dr. Bailey, I definitely want to hand this part over to you, but thank you all for joining us. Now, if in fact you don't see one of your friends here, your and partners for your community, if you are a partner here, you have a CBO or your IPCA, I'm challenging you right now to share this with someone, because one of the biggest things for the consortium is that our voice be heard. We talk about it all the time, but in order for that to happen, we have to make sure that the voices are in the room. Now, this is a StreamYard, so you're going to be on Facebook Live. It's not regular Zoom, so you will not be able to unmute. But questions, if you have questions, put them in the comments, and our team on the back end will be sure to get those questions to the panelists. But ensure that if you do not see somebody on here that should be on here, send them a message and let them know, get on now, because we're about to have a really good conversation with Fulcrum. And
All right.
Bailey.
Thank you much. Thank you so much for that, Sharonda. Let them know, you all. I see so many of you that have joined us directly. We do ask that you go ahead and register if you ask them to join us, so that we can collect that info, know who's here, who's interested in these types of meetings. But join us on Facebook. We're following, just like Sharonda said. Now, it is 6:10, and I'm going to jump right on in. So as you all know, hello everyone. Let's go there. I'm Dr. Lakia Bailey, one of the founders and executive director of the Sickle Cell Consortium. As you all know, we are a network of community-based organizations throughout the country, and now with our addition of our international partners throughout the world. One of our primary and one of our major pillars surrounds education. We approach education in three ways: in print, through our resources and patient-powered educational programs and booklets; online through Warrior University; and in person through things like the Warriors Convention. Part of the online network of Warrior University are also these town halls. We do them throughout the year. This is our final one of the year. I can't believe it's already December. One of the main purposes of partnering with industry, we've done many different types of town halls, from talking all about marijuana and the legislation around it and the do's and don'ts and what doctors have to say, what patients and caregivers have to say about it, all the way through to gene therapy and editing with multiple. One of our reasons for doing this and this type of town hall, like when we partner with industries such as Fulcrum Therapeutics, these pharmaceutical companies, is to ensure that our sickle cell community is fully aware of all of the options. Who are the players here? Who are the stakeholders? What industry is doing? What is academia doing? What is government doing? Which is why we've done some of these things with CureCI, NIH, FDA. We did an entire series, a 3-class series with the FDA, these types of things, so that our community is informed. You cannot have informed consent without first being informed. And that is the purpose of these meetings, to ensure that you all know who's out there, who's doing what, and to give you all the opportunity to ask the difficult questions. We have been doing this for many years now, and this is an incredibly intelligent community. You all know all the questions you want to ask, and we want to give you a platform to hear, to learn, and to ask those questions. One of the things I do want to ask you all as you're typing and chatting and communicating in the chat, one of the important things that I really like to know is, where do you get your information from when it comes to clinical trials, when it comes to new drugs and new things that are on the market or coming on the market, studies that are coming? Do these town halls help you? Do you get your information from your provider? Do you get them from other warriors and other caregivers? Where do you get this information? I'd love it for some of you to type that in the chat and let us know, where does this come from for you? What is the best way to learn? How can we assist? We have different, of course, you may have seen them. If you've come to any of our events, you've probably gotten them over and over again in the mail, all the different publications that we've created, lots of the educational materials, all of that. But where does this come from for you? Where do you want to learn? Where do you get the most information? Please go ahead and type it in the chat. We're following along, those of you watching from YouTube, from Twitter, or Facebook as well. And I would love to know that information. Now, the purpose of this is to bring to you Fulcrum Therapeutics. Now, as full disclosure, sometimes our town halls are hosted in collaboration with industry, such as Fulcrum, and we disclose this to you. Sometimes it is entirely led by one of our partners, such as when we did some of the other ones in the past, like when we brought Bluebird and Vertex up. Sometimes they're run entirely by us and they are unconnected to industry. This town hall is connected to Fulcrum Therapeutics. Nothing you hear today is meant to replace the advice from your physician. This is, however, nor is it meant to reflect the opinions of the Sickle Cell Consortium or any of our partners, our staff, or our board. This is an opportunity for you to learn, for you to hear, and for you to ask the questions that you wish to ask. Fulcrum Therapeutics has been a sponsor with the Sickle Cell Consortium many times over the years, and we are grateful to have been able to connect with them and to learn, and for their support in our many initiatives. So without further ado, I am going to bring up Danielle Branch. Danielle with Fulcrum Therapeutics. And we're getting some really wonderful comments in the chat, Danielle. My favorite so far is, where do you get your information? The Sickle Cell Consortium. Love that. Some great things in the chat, Danielle, about where this info comes from.
Absolutely.
And it sounds like quite a few of them get it from things just like this.
This is
Hi
awesome.
there, Danielle.
Good evening, everyone. Thank you, Dr. Bailey, Dominique, and Sharonda, and the team at the Sickle Cell Consortium. I'm so excited to be here this evening and really, truly honored to be one of the representatives from Fulcrum Therapeutics speaking with you this evening. My name again is Danielle Branch. I am actually Associate Director of Patient Affairs at Fulcrum, and really what my role is to be your representative at Fulcrum. My job is to make sure that your voice is heard, and not only heard, but also implemented throughout our company, and making sure that you are informed and we're informed as well as Fulcrum about the sickle cell community from the perspective of caregivers, warriors, advocates in the community as well. So I like to be a little bit informal. So I want to make sure I check Sharonda's box. She asked me, you have to give a shout out of where you're from. So I am actually in Atlanta, but by way of Philadelphia. I was in the comments looking at some individuals from Philadelphia. So I want to make sure I give that shout out as well to my home team and Go Eagles as well. But before we kind of get into our presentation, I really wanted to make sure that I truly express to you that we are truly honored to be here, and that this town hall is really supposed to be a dialogue. As Dr. Bailey said, we want to make sure that you're informed about who we are as a company and who we are as individuals. I also like to say that this is kind of like our first date. Like any relationship and partnership, we have to build trust. We have to talk. We have to be open and honest and transparent throughout this process. And I like to think this is our first date. And you'll get to meet some other of my colleagues and some of the leadership at Fulcrum to kind of start that dialogue. So please be open and honest, be engaging with us, ask tons of questions, because this is exactly what we're here for. So without further ado, I would like to introduce our CEO and president of Fulcrum Therapeutics, Alex Sapir.
Right, Danielle, thank you. Thank you so much. And Sharonda and Dr. Bailey, I cannot thank you enough for this opportunity. I am absolutely just delighted to be here with all of you this evening, albeit virtually. As the comments were coming through and the various names, I saw a couple of names that I actually knew. And these are a couple of people that had come up to me and that I had had the opportunity to meet about 4 or 5 weeks after I joined the company. And they asked me, why did you join Fulcrum? And the answer that I gave was the same answer that I gave 35 years ago when I decided to join the healthcare industry. And that's really to make a difference in patients' lives. I think what's really unique about rare diseases like sickle cell is that the work that we're doing does make a difference, but it truly has the potential to be transformational in people's lives. And I think that's why we're so excited about some of the work that we're doing at Fulcrum today. And you'll hear from Dr. Engelman in a couple of minutes about some of the early work that we've been doing in this product called pociredir. And we truly believe that with some of this early stage data and our ability to increase levels of fetal hemoglobin up to the levels of the high 20s or low 30s, we believe that this can be truly transformational for patients. There's still a lot of work to be done. And the only way that we can make that vision a reality is with all of us, with us here at the company, working on the study, with the physicians in the centers enrolling, and most importantly, patients making the decision to really enroll in these studies to really help advance the field even further. So I can't be more excited about what the future holds for all of us in terms of realizing this vision. It was funny, before I joined Fulcrum, I had an opportunity to get connected with Ted Love. I'm sure many of you know Ted. He was the former CEO at GBT. And I asked Ted very specifically, I said, Ted, what is it going to take for me to be really seen as a true partner amongst the sickle cell community, and in particular the warriors? And he basically said, Alex, you need to do two things. Number one, you need to show up. And number two, you need to listen. And I remember he specifically said, God gave you one mouth and two ears for a reason, and you should use those ears to listen. So I'm here. You're going to be seeing a lot more of me in the years to come. And I'm just really looking forward to spending the next couple of hours with you and just listening and absorbing as much as I can. So again, thanks so much for the opportunity. And Danielle, I guess I'll turn it back over to you.
Yes, thanks, Alex. This is thank you for that introduction. We're going to go to kick it off to Mel to let him say a few words and introduce himself to the community as well.
Yes, hello everyone. I think by now I've met many of you at whether it was Cayenne meeting in California or a meeting down in Houston. But again, Mel Hayes, I am the Executive Vice President of Patient Experience here at Fulcrum. It's a new function which was newly created less than 2 months ago to specifically focus on the patient experience. And it's our goal to ensure that we are as closely aligned as possible with the priorities of the sickle cell disease community. And later on, Danielle will share with you more details about our function, how we're set up. But really, it's a tremendous opportunity for us to, at the early stage of our development as a company, to really over-index on the experience. I've been a sickle cell for about 10 years off and on, but primarily on the curative side. And that was great, exciting, and so forth. But what really got me excited about Fulcrum was the opportunity to potentially bring forth a small molecule once a day disease modifying therapy, an HbF inducer. Because I really do truly believe that this type of therapy can transform sickle cell disease globally. As we know, in the United States, about 110,000 people in the United States have sickle cell disease. But globally, it's in the millions. It's in the millions. And I feel it's incumbent upon us as a small biotech to do whatever we can to move forward this program and other programs like ours to ensure there are options out there for the community. I think that if we do our job right the next several years, and we're pretty early, as Alex mentioned, we're going to have a chance to truly transform the African diaspora. I can't tell you how excited I am. I mean, this is a tremendous opportunity for us. We want to show up and be there for you. We want to listen. As Alex mentioned, it's critically important for us to do that. And we're looking forward to continuing to dialogue and work with you. Next slide. Okay, so some of you may have asked, what is Fulcrum's mission? Or you want to know what Fulcrum's mission is. Go to the next slide, Danielle. Fulcrum's mission is really simple. It's to treat the root cause of rare diseases. Who are we? Well, we're a company that is solely dedicated to co-creation. And what does that mean? We take it seriously that whatever we produce, we do with the community, with the patient community. We ensure that as we develop materials, initiatives, programs, that we're constantly engaging the community in the right way. So my commitment to you is to ensure that whatever leaves our organization is actually vetted by or seen by a warrior. That's our commitment to the community as a whole. We aim, though, as a company, to deliver disease-modifying small molecule therapies. That's what we're here to do. Obviously, Pociredir is our first shot on goal. We feel very excited about it, but we're going to continue to look at ways in which we can innovate and move forward our organization and to move forward the sickle cell community in a way that's bringing value to each and every single one of you. Now, in a few moments, you're going to hear from Dr. Bill Engelman. Recently, we just started back our phase 1B program called the Pioneer Study. Now, the Pioneer Study, in my I guess you would say, humble opinion, the name is super special. Why? Because it was named by a warrior. We're a small company, less than 100 people. We had about 85 people submit names. And after all of that, those names were cast through a democratic process where every vote was counted, we were actually able to choose Pioneer as our study name. So it means a lot to us to have this as our first real branded study name, the Pioneer Study. And it makes us proud that this came from a warrior. For those of you who I saw at the meeting in Cayenne and also the meeting in Houston when I attended and said a few words, you may remember me saying something on stage along the lines of, we're small and we're mighty. We truly feel that it doesn't matter the size of the organization. If the heart is there, if the mindset is there, if the passion is there, and if the cooperation is there, we're going to have an opportunity to make a huge impact on the community. And we're looking forward to partnering with you along that journey. Now, right now, I'm going to turn it over to Dr. Bill Engelman, who's going to share with you some more details about the program and the asset Pociredir. Dr. Engelman, turn it over to you.
Hi, I appreciate it. Hi, I'm Dr. Bill. Thank you, everybody, for allowing me the chance here to speak to you tonight about Fulcrum as well as Pociredir and our Pioneer trial. But let me talk a little bit about why I came to Fulcrum and why I'm so excited about working on Pociredir in this clinical trial for sickle cell disease. As Mel said, Fulcrum, we're small but mighty. We have a team here that's very committed and passionate about moving forward. And I love that about this group. But why did I want to come into here to this trial? Why do I want to, work on sickle cell disease? In order to understand that, I got to tell you a little background about myself. And I don't want to talk too much about this, but I have a chronic disease I've lived with my entire life. I've had several friends of mine that have been lost to the same disease. Well, it's not sickle cell disease. I feel like there's some overlap here. I've been privileged to be able to get the health care that I need to not only survive, but thrive. But here's the problem with that. Healthcare should not be a privilege. It should be a right, and it should be a right for everybody. Everybody should be able to get the appropriate healthcare that they need. What I'm seeing here with this drug, pociredir, I'm seeing that this drug has the potential to be a game changer for sickle cell disease, and has it's a once a day pill that can be taken and helping so many people with sickle cell disease, and it does not require a whole lot of resources to get. And I'm seeing that this can be, like I said, help so many people around the world with it. With that said, I'm going to move in and speak about our clinical trial. So can I go to the next slide, please? Next slide, please. So first, I want to start with talking about what's happened to us over the past year. We did have to go on clinical hold. What does that mean? So I'm going to read this here word for word. A clinical hold is an order issued by the FDA to the sponsor of an investigational new drug application to delay or a proposal, clinical trial, or to suspend an ongoing clinical trial. What does that mean? So the FDA came and put us on clinical hold, and there's many reasons why they can put us on clinical hold. For us, it had to do with some safety that they saw in the animal studies that we had done. Next slide, please. So what it's seen, we have done as a normal routine for research, we had done many studies with animals that had taken pociredir. And in those animals, a small proportion of those animals had some blood cancers, otherwise known as hematological malignancies. So they saw it in a small portion of those animals. What they also saw was in a drug that has a similar biochemical process to pociredir. The drug is called tazemetostat, and I'll speak a little bit more about that later on in this presentation. But in that drug as well, that was given to cancer patients, not sickle cell patients, but cancer patients, they saw a small proportion of those patients as well that had developed cancer. Now, those patients that took this drug, tazemetostat, they had cancers prior to this and took multiple therapies that were not effective or did not work. They did not work. And tried these therapies as well. So they had other problems going along with them, but they did develop some cancer. To date, we have not seen any cancer in any humans that have taken pociredir. However, due to what we have seen in the animal studies, as well as what was seen in tazemetostat, we want to make sure that we're moving forward in the safest possible manner that we can. So we have worked with the FDA to make sure that we're going to move forward in the safest way possible. Next slide, please. So how are we moving forward in a safe manner? Well, in order to understand that, let's talk first about clinical trials and how they're conducted and why. So as you can see here, clinical trials are conducted to see if a drug is effective in humans. How well is it doing in humans? And how much benefit is it giving the humans that it's taking? So for example, a drug that will lower cholesterol, give a drug for cholesterol, is it lowering the cholesterol? And how much is it lowering it by? A drug for diabetes, is it, lowering blood sugar? And how much is it lowering by? So clinical trials help to answer these questions. What they also do is they help to find out the risks of that drug. Now, all drugs have risks. Heck, some of the foods we eat have risks, like, if we eat too much sugar or eat too much fat, those all have risks. But all drugs certainly have risks. One of the things we do in a clinical trial is to find out what those risks are and how serious they are, and if there's anything we can do to mitigate those risks. So in order to make sure that the benefits of taking that drug outweigh any of those risks. So we have to have an appropriate benefit-risk balance here. I also want to mention one other thing about clinical trials and speak about the types of clinical trials that need to happen in order to get a drug on the market. There's phase 1, phase 2, and phase 3 clinical trials that are required to get a drug on the market. A phase 1 clinical trial focuses on safety, on the safety of a drug. Usually they're very short. They can last, a few weeks to a few months. They have very few patients, maybe 20 or 30. And again, like I said, they focused mainly on safety. a phase 3, I'm going to jump to a phase 3 clinical trial here. A phase 3 clinical trial focuses on whether or not the drug is effective. Is the drug doing in humans what it's supposed to do? These trials are much longer. They last up to about a year or more and require many, many more patients, 100 plus patients. Now, with that said, I said it focuses on how effective the drug is, but that doesn't mean we're not looking at safety. We're still looking at safety in a phase 3 trial. Similar to the phase 1 trial, while it focuses on safety, we're looking to see if it's doing what it's supposed to do in patients. We're still looking at that, but it's just not the main focus of the trial. In this stage of pociredir, we are currently in a phase 1 trial. This is first in human studies. And we want to make sure that we're moving forward as safe as we can in this phase 1 trial. And that way we can make sure that the drug is safe for everybody. So our clinical trial, this Pioneer trial, has since been open based on what we have done with the FDA to make sure that it's safe. And how did we do that? So what we did was we are bringing in patients eligible to enroll in this clinical trial that have the following 2 items here at the bottom. So these are patients that have developed or have a high likelihood of developing severe events related to sickle cell disease. So these could be patients that have many pain crises. These could be patients that have end organ damage based on their sickle cell disease. Or these are patients that could have any other known events from sickle cell disease, such as acute chest syndrome. Also, we're bringing in patients that have tried other therapies for sickle cell, but those therapies have not been effective and there's no other options for them. So these are the patients that we're going to be bringing into our trial to make sure that we move forward in a safe manner. Next slide, please. But now let's talk about how our drug works. In order to understand how our drug works, we have to talk about how, what sickle cell disease and how that disease is done. And as I'm sure all of you know, sickle cell is a disease of red blood cells. And those red blood cells are misshapen or misformed. They turn in that classic crescent shape or that sickle cell shape. And why do they do that? Well, the reason why they do that, if you take a red blood cell and open it up, it has a specific protein on the inside called hemoglobin. The job of hemoglobin is that it goes to lungs, picks up oxygen, and it delivers that oxygen to cells in the body so those cells can use it as energy. Now, I'm going to be talking about 2 different kinds of hemoglobin here. The first one is adult hemoglobin, and the other one will be fetal hemoglobin. But let's first talk about adult hemoglobin. So in a red blood cell, there's millions of these adult hemoglobins. And what happens in sickle cell, I'm sure some of you know this, is that those hemoglobins, stick together. They bind together. Some people like to say they form chains. No, they don't form a chain. What they end up forming actually is these long, stiff rods. And when they form these long, stiff rods, what that does is that deforms the red blood cell, turns it into that sickle shape. And when it turns into that sickle shape, that's when all the problems occur, right? That's where the pain occurs. That's where some of the end organ damage can occur. And sometimes these red blood cells break open and release some of the toxins that are held within the red blood cell. It releases it into the bloodstream and causes problems there. So how do we fix that? We can fix that by producing another hemoglobin called fetal hemoglobin. And it's been shown if we can get enough fetal hemoglobin, about 30% or more in red blood cells, that prevents that adult hemoglobin from binding together and forming those rods. And it keeps those red blood cells from deforming. So how do we produce fetal hemoglobin? Well, here's the interesting thing about fetal hemoglobin. We've all produced it at one point in our lives. As the name implies, fetal hemoglobin, we produced it when we were fetuses, when we were in our mother's belly, when she was pregnant with us. Fetal hemoglobin gets shut off not long after we're born, but that machinery to make it is still there. So how do we turn that machinery on? Well, it's actually a 2-step process here to turn that machinery on. First, we have to build the actual machine to make the fetal hemoglobin. And then once that machine is built, that then makes the fetal hemoglobin that can be put in the red blood cells. And that's what our drug does here. If you look here on the left of this slide, this PRC2 complex, this blue thing over here on the left, what this guy does here, okay? Well, let's look at the bottom here first. At the bottom here, this is our DNA. And our DNA here is shut off by these little red clovers that you see here. That's called a trimethylation. But this PRC2 complex, what it does is it goes and shuts off the DNA at this location. This location is where the blueprints are to make that, put together the machinery to make fetal hemoglobin. What our drug does is it binds, it grabs onto this PRC2 complex here at the specific site. We call it the EED site. It grabs onto it, and it prevents that complex from locking up that DNA. So when it prevents it from locking up that DNA, what that DNA can do is open up, it gets read, the machinery to make the fetal hemoglobin then gets put together, and the fetal hemoglobin gets made. It's a beautiful process. Now, how do we know that this happens? Well, we did see this in tests that were done in test tubes, and we also saw this in animal trials that were done. So pociredir was given to animals, and we saw an increase not only in this machinery, which is called HBG mRNA, but we also saw an increase in fetal hemoglobin. We also have been seeing it in humans as well. Next slide, please. So this is data here from our healthy volunteer study. So this was given to healthy people to see how well pociredir worked. If you look at the graph on the bottom, you look on the left over here, HBG fold induction is over here. And on the bottom, it's the number of days that these healthy volunteers took pociredir. Now, this HBG fold induction, this is measuring the activation of that machinery I told you that produces fetal hemoglobin. And you can ask me, Dr. Bill, why aren't you measuring fetal hemoglobin? Why are you measuring the machinery instead? Well, it's a great question. The reason why, I'm going to use this analogy. So we talked before about where we're from. I'm from the city of Detroit. I grew up around Detroit, where the automobile industry, that's the Motor City, Motown. We make cars there. Now, in the Motor City, we have all these plants that make cars. And in the back of the plants, there's a parking lot where they put all the new cars in, and it fills up with all these new cars. Now, imagine we have a brand new automobile plant, and we just now turned it on, okay? If we just now turned it on, and we go up back to look at the parking lot, will there be cars there? No, not at this point. It's going to take several days to weeks, even months, to fill that parking lot up with cars. Well, it's similar here. It's a similar analogy. What we're seeing here is the machinery get turned on, but it's going to take several days to weeks before we see that fetal hemoglobin being produced. So we only had these healthy volunteers for 14 days, so we had to figure out if it was working. So we ended up measuring the machinery, the activation of the machinery instead. And we did see what I'm calling and what we call a dose dependent study here. So what I mean by that is that if you can see here, going up from 2 milligrams all the way up to 30 milligrams, we're seeing an activation, slight activation at 2 milligrams that jumps up when we go up to the 6 milligrams, that jumps up even further when we go up to the 10 milligrams. And then we go up to the 20 milligrams, it jumps up even further. Now you can see here too, that it barely jumps up when we go up to 30 milligrams. You ask me, Dr. Bill, 30 milligrams, it's more there. Why is it not working as much at the 30 milligrams? Well, I'm going to go back to my car plant analogy. Imagine you open up an automobile plant that's producing cars. You bring in a few workers in there, 2 workers in there. They're barely going to make any cars. You bring in more. They're going to make more and more and more. Well, there's going to be a point there where you have so many workers in the plant and not enough tools and not enough machinery to produce, more cars. And there's a maximum of cars you can produce. Well, it's similar here. The body has a maximum amount of fetal hemoglobin that it can produce. And we're seeing that maximum amount at 20 milligrams. We're also seeing production of fetal hemoglobin in sickle cell patients. Next slide, please. So here's data from our current phase 1b trial. And this trial is in sickle cell patients. And what we're seeing on the left over here, this says absolute change in fetal hemoglobin from baseline. What that is that's a measurement of the amount of fetal hemoglobin that's being made by pociredir. And then down here at the bottom, you say days on study drug, days on pociredir. This study went out for 12 weeks. So patients are on the study drug for 12 weeks. So at day 84 here, you'll see a dashed line here. That's when they stop taking pociredir. That's when warriors stop taking pociredir. And as I mentioned before, too, in healthy volunteers, it's similar here that this is dose dependent. So we see in 2 milligrams after 12 weeks that they produce about 2 to 3 percentage points above baseline with their fetal hemoglobin. It increases as we go up to 6 milligrams. You can see that's around 8 to 9%. But in the 12 milligram dosing here, right, you can see it's going up pretty fast, and it goes up to almost 10, 11 percentage points after about 4 to 6 weeks. Unfortunately, this is where we went on clinical hold, was here at the 12 milligram dosing when patients were there. But if you can imagine, and imagine this line going out, extending way out, how far up this could go. If patients have about 25 to 30% or more fetal hemoglobin in the sickle cell, in their cells, that prevents them from sickling. So if you can imagine, if we can get up to the 20% range, and this is in addition to what patients make, without any drugs, we can easily get into that 25, 30% range. And we're very excited about this because this data is showing that this is going to be very effective. Next slide, please. So this is our clinical design. This is what we're doing to date. Our study population, you can see over here on the left, that we are looking for sickle cell warriors ages 18 to 65, adult warriors. It's an open label. So what that means is that all patients are taking pociredir, and we do not have placebo on this clinical trial. We also are not allowing patients to have hydroxyurea. That's in response to the safety, and I'll speak more about that in later slides. But patients cannot be on, or sickle cell warriors cannot be on hydroxyurea for this study. The study design is a dose escalation where we're looking for 10 patients per cohort. So what that means is we're looking for 10 patients to take each of the dosing levels. Screening period of 4 weeks. What that means is that a patient comes in to see their doctor. The doctor will ask questions. The doctor would maybe do some exams and tests. And if they're eligible, they can enroll in our clinical trial. And once enrolled into the clinical trial, there's a 12-week treatment period for pociredir. Once that treatment period ends, we will continue to follow the warriors for up to 4 weeks. And that's in order to look and to make sure that there are no other safety issues involved with the drug, even up to a month after. Now you can see here at the bottom, this is who we've enrolled and who we're looking to enroll in our clinical study. To date, we've enrolled 16 patients. That's been 10 into the 6 milligram. We only enrolled 2 in the 2 milligram dosing because we just wanted to see it was pharmacologically active, and it was. We also enrolled 4 in the 12 milligram. As I mentioned before, it's because of the clinical hold that those patients had to stop the 12 milligram dosing. We are looking to restart at the 12 milligram dosing, and we are restarting 12 milligram dosing, looking to bring in 10 patients, 10 warriors, who can take 12 milligrams over a 12-week period. Once we get 8 patients into this 12 milligram cohort, what we're going to do is to look at the safety as well as other labs from this study. If everything looks good and it's safe, we're then going to allow patients to take 20 milligrams. And we're looking to get 10 patients to take 20 milligrams in that cohort. Next slide, please. So this is what we're looking for, and these are the endpoints. As I mentioned before, in a phase 1 trial, safety and tolerability are the main endpoints we're looking for. So that's what we're looking for. That's the primary endpoint we're looking for, is safety and tolerability. So that includes any adverse events that may occur while taking the drug, any changes in laboratory values. The doctors who you will go see on this trial will do physical examinations to see if there are any changes there, as well as any vital signs. And we'll be looking at the conduction of the heart. So we'll be looking at the electrocardiogram of heart. And these are standard measurements that are taken for just about all phase 1 clinical trials to make sure that the drug is safe. What we will also be looking at as a primary endpoint is the PK parameters. What the PK parameters stand for is that when you take a drug, it's going to go into the bloodstream, and we need to see how much of it ends up in the bloodstream. What are the concentrations we see in the bloodstream, and how long it takes before it exits the system, exit the bloodstream. And that's what these PK parameters do for us, is that they measure the concentration in the bloodstream, as well as how long it takes to get out. Then we also have some secondary endpoints. We'll be measuring the fetal hemoglobin levels shown here, and we'll be measuring how healthy the red blood cells are by looking at what's called unconjugated bilirubin. With red blood cells, when they sickle, they can break apart very easily and release, like I said, toxins in the bloodstream. One of those is bilirubin. So we are looking at the bilirubin levels to make sure that the red blood cells stay healthy. And then you can see some of the exploratory endpoints here on the right. The number of vaso-occlusive crises. We're also going to be looking at what's called F cells. And this is an important point as well. So I mentioned before, we're looking to increase fetal hemoglobin. But what we also want to do is make sure that we're increasing fetal hemoglobin in all the red blood cells. So let's say, for example, that the fetal hemoglobin gets increased in only about half of the red blood cells. Well, the other half now don't have protection, and those can sickle as well. So what we want to do is make sure that when we produce the fetal hemoglobin, it's in all blood cells. And when it's in all blood cells, there's a term we use called pancellularity. So that's what we are looking for, is pancellularity in these red blood cells. And we're looking for other things as well, other markers for sickle cell disease. Next slide, please. Now let's talk a little bit about the safety that we've seen so far with pociredir in warriors on this Pioneer study. To date, we've had 16 warriors that have entered in the study and have seen 19 treatment emergent adverse events in 10 of those warriors. And we've seen 4 of those being related to the study drug, to pociredir. Now, I want to step back here and talk about relatedness to study drug. When a study drug gets given to patients and people who come into a clinical trial, the doctor that sees these adverse events has to give a relatedness to it. And it can be either definitely related, it can be probably related, it can be possibly related, it can be probably not related, and definitely not related. So those are the categories they give for relatedness. We did see 4 related events here. They were tinnitus, which means ringing in the ears, intermittent lip numbness, diarrhea, and headache. They were all considered possibly related, and they were considered possibly related because they were started not long after taking pociredir on the trial. However, just note, all of those events resolved while the warriors were still taking pociredir. So to date, we have not seen any signals or any other safety concerns. It's been rather unremarkable. I also do want to mention what we also have not seen is something called myelosuppression. What myelosuppression means is that the bone marrow produces all the blood cells. They produce red blood cells. It produces also white blood cells, which are used to protect us against infection and other things. And it produces platelets, and platelets are used for blood clotting. So it produces all of those. What myelosuppression means is that those blood cells fewer of them get produced because of some kind of toxin in the bone marrow. What we see with hydroxyurea is that there is myelosuppression with hydroxyurea. We are currently not seeing that with pociredir, and that's a big plus for this drug. Next slide, please. So again, this is why we had to go on clinical hold, and I spoke a little bit about this earlier. You know, we had seen some cancer that were in the animal studies that were done, as well as within a drug that has a similar mechanism of action. And that drug is tazemetostat. Now, what do I mean by similar mechanism of action? Well, remember I told you before about the PRC2 complex and how pociredir binds to it and prevents this complex from putting those locks on the DNA. Tazemetostat drug also binds to this PRC2 complex and blocks it as well. But that's where the similarities end for our drugs. The reason why I say that is because with pociredir, okay, it never fully blocks this PRC2 complex. In other words, there's several of them around, and it blocks it at about 70 to 80%. So there's still 20 to 30% that is active. Tazemetostat blocks it 100% of the time. 24 hours a day, it blocks it. And as I mentioned before, it is used for cancer patients who have already used other therapies for their cancer and have other risk factors associated with cancer. But because of these risks, like I said, we want to make sure that we're going to move forward safely in this study. And we work with the FDA to make sure we balance out the benefit risks so that the benefits outweigh the risks. And we have since been off clinical hold since the 18th of August of this year. Next slide, please. So what we had to do to get off clinical hold is that we had to do these key elements here. Patients that have previously used hydroxyurea and at least one of the other drugs on the market for sickle cell disease. So those other drugs are voxelotor, also known as Oxbryta, crizanlizumab, also known as Advetco, as well as aglutamine, which is Endari. So patients have had to use one of these and have been non-responsive or intolerant to one of these medications. Also, when we originally opened this study, we called it an all-comer study. So patients that were well controlled and did not have any pain crises or vaso-occlusive events were allowed in this study. That has since changed. We now have a more severe patient population that will be eligible for the study. So these are patients that have more pain crises or vaso-occlusive events and or have end organ disease because of the sickle cell disease. We also are excluding patients on hydroxyurea, and I'll speak a little bit about this now. There's a warning on the hydroxyurea label that it causes cancer. Now, in speaking with many doctors and hematologists across the country, as well as in many studies that are in the literature, that risk is very, very small to none at all. That warning has been on the hydroxyurea label for several decades. It started on there when hydroxyurea was used as a cancer drug, and it's never come off since. But it's been shown that hydroxyurea does not cause cancer in sickle cell patients. However, like I said, because we want to move forward in a safe manner, we are going to exclude patients that are on hydroxyurea at this point. Next slide, please. So what are the inclusion criteria? Here's what we have for the inclusion criteria for the hydroxyurea use. As I mentioned before, patients have had to use hydroxyurea but have been non-responsive or intolerant based on any one of the following criteria. And they only need to meet one of these criteria, not all of them. So for the first one here, if patients have pain crises or vaso-occlusive crises while being on hydroxyurea for 6 months, they have the same number or higher from baseline, that's considered an inclusion criteria. If patients have acute chest syndrome, which is a known problem with sickle cell disease, if they have acute chest syndrome while being on hydroxyurea for at least 6 months, that would also be an inclusion criteria. If patients cannot tolerate hydroxyurea, defined here, either having a severe allergic reaction to it, having any of the hematological toxicities. Remember I mentioned before, the myelosuppression that hydroxyurea has. So if they have any of those, and there's a decrease in red blood cells, white blood cells, or platelets, that would also be considered an inclusion criteria for this study. If patients have a known gastrointestinal side effect from hydroxyurea, such as nausea, vomiting, diarrhea, or abdominal pain, that would be an inclusion criteria for the study. If they have the inability to tolerate any of the other side effects that are known for hydroxyurea, so as they're listed here, fatigue, dizziness, headache, fever, leg ulcers, and or skin changes, again, that would be an inclusion criteria. And then these last two inclusion criteria are general for just about any drug regardless. Unmanageable drug-drug interactions and patient refusal. So a drug-drug interaction would be if a sickle cell warrior is on a different medication for a different reason, and if taking hydroxyurea causes all kinds of bad side effects because they're taking that other drug and have to stop it, that's an unmanageable drug-drug interaction, and that would be an inclusion criteria for this study. Next slide, please. What about the other drugs? Voxelotor, crizanlizumab, or L-glutamine. So similar to hydroxyurea, there has to be non-responsive or intolerance after being on it. So for voxelotor, it's indicated to increase hemoglobin by at least 1 gram per deciliter. That's what's in the label. So if that cannot be reached while being on voxelotor for at least 6 months, that would be an inclusion criteria. For crizanlizumab as well as L-glutamine, similar to hydroxyurea, it's indicated to decrease pain crises or other vaso-occlusive events. So if a patient's on it for 6 months and they have the same amount of pain crises or more, that would be an inclusion criteria. And then these last ones are similar to hydroxyurea: inability to tolerate any of the adverse effects of these therapies, unmanageable drug-drug interaction, and patient refusal. But I want to highlight this second to last one here, the lack of access to therapy. What we're seeing and what we're hearing about these drugs, first off, they're very expensive. And many times payers and insurance companies do not want to pay for these drugs because they're so expensive. They're very difficult to get. And sometimes they'll make warriors have to jump through hoops in order to get these medications. So that would be lack of access to this therapy because they're unable to get them due to the payments, as well as due to the high prices, as well as those hoops that warriors have to jump through. And with regards to crizanlizumab is a once-a-month medication where warriors have to come into the clinic and get an IV infusion. And there could be all kinds of reasons why that may be difficult. Employment reasons, childcare, many other reasons, transportation reasons why it may be very difficult to get on crizanlizumab. So that could also be considered a lack of access to therapy and would be considered an inclusion criteria for this study. Next slide, please. So now we've also had to bring in patients that are warriors that are more severe. And you see all these inclusion criteria for the severity of the disease. And warriors only need to meet one of these criteria in order to get into the study. But I like to bucket these into two different categories. The first three bullet points here are for those acute events that occur with sickle cell disease. Then the last three bullet points are end organ damage caused by sickle cell disease. So the first one here, greater than or equal to 4 episodes of sickle cell pain crises over 12 months or 2 over 6 months. And I want to define what pain crises is for our study. What a pain crisis is for us is that if a sickle cell warrior needs an additional pain medication other than what they are prescribed at home. And that could mean that they go into an emergency department or they go into the hospital or have to run the clinic. It could also mean that if they call their doctor and ask for an additional prescription because they're having such great pain at home, that would also be considered a pain crisis that would allow them into the study for them. Also, any of the other known complications from sickle cell disease, such as acute chest syndrome, hepatic, which would be liver or splenic sequestration, or priapism. If one of those events occurs over 12 months, then the number of pain crises gets cut in half. If 2 of those events occur over 12 months, it doesn't matter, regardless of the pain crises, the sickle cell warrior would be eligible for this trial. And then these last 3, these are the end organ damage that is caused by sickle cell. Pulmonary arterial hypertension, as defined here, which a doctor will diagnose using this criteria. Chronic kidney disease, which your doctor would diagnose using this criteria. And then this last one, it says meets medical criteria to receive but are contraindicated for chronic transfusions. This would be, for example, for patients such as patients who have had stroke and need a chronic transfusion due to their stroke, but otherwise can't get it because of allergic reactions, alloimmunization, or other transfusion reactions. Those patients those warriors would also be eligible for this trial. Next slide, please. Now, finally here, I want to speak about the risk that was seen with tazemetostat in their clinical trials. As I mentioned before, tazemetostat has a similar biochemical process or a similar mechanism of action to pociredir, but it's also used for cancer patients. It's used for epithelial sarcoma as well as follicular lymphoma. What they had also seen in their animal data was that they had a 10% risk of lymphoma that was seen in rats. What they had seen in their clinical trials across 822 patients being on it for at least 6 months was that there is a risk for a secondary malignancy in 0.7% of the patients. And those are all listed here. Now, I want to emphasize that all of these patients had been on either prior radiation or prior chemotherapy prior to getting these secondary malignancies. So they had other risk factors that may have contributed to those malignancies. And you can see all of those secondary malignancies are listed here. There were 2 patients that had myelodysplastic syndrome, 3 that had acute myeloid leukemia, and 1 that had a T-cell lymphoblastic lymphoma. If you remember, I mentioned that in the animal studies, lymphoma was the cancer that was seen in the rats. Only 1 lymphoma was seen here. So we're not sure how to interpret the animal data when it comes to what we see in humans. I also want to show how long patients were on tazemetostat before they developed these secondary cancers. For the fastest one was this T-cell lymphoblastic lymphoma that occurred in a 9-year-old pediatric patient who was also on prior radiation and prior chemotherapy. And that occurred 61 weeks after being on the drug, or 14 months. The second fastest one was the myelodysplastic syndrome that occurred 66 weeks in a patient who was also on prior radiation and prior chemotherapy. Our drug for this phase 1 clinical trial is only going to be 12 weeks long. I do have to say this too. We believe that our risk for cancer is less than 0.7%, but we're not sure how much less. That's why we want to make sure that we move forward carefully, safely, and slowly for this clinical trial. Now, I want to thank you very much for allowing me to present this to you, and I hope that we can meet again in the future, and we can provide a significant healthcare and therapy for sickle cell patients. And with that said, I'm going to hand this off back to Danielle.
Thank you, Dr. Bill. What a great presentation. So much information. So I'm going to provide a little bit of a commercial break, let everybody process all of that great information that Dr. Bill just presented, and want to make sure that I acknowledge all the great comments in the all the questions in the comments, that we will definitely get to those right after I talk a little bit about, one of the things that we talked about earlier is that really our commitment to the community, and what Mel, as far as he's over our patient experience department, and what patient advocacy, our community engagement and partnership really does look like here at Fulcrum. So next slide. Next slide. Is really want to talk about our commitment, because we wouldn't be here without you. And all the information that we just Bill presented about Procedure and our clinical trials is not going to be made possible without you and being part of the community. And part of that is really establishing, our relationship and building that trust. And that, as you see here on the screen, we really want to make sure that we build lasting partnerships in the community. And what that really does mean to us, like I said, is making sure that we have open and honest and transparent dialogues, and having these opportunities to talk to you tonight at the town hall, and also really understand your experience from a warrior's perspective and a caregiver perspective, because we want to make sure that you are heard, and that the information that you give us in this dialogue, that we really truly take it to heart, and that we will actually implement those suggestions and feedback and answer all those questions in the comments here. We really are truly here to listen and learn from you and be true partners, and also develop impactful programs. The town hall is a perfect example of making sure, as Dr. Billy said earlier, is to make sure that you are informed. Not only you are informed about the information we're presenting to you, but also we're informed about the information you want us to know about as well. So next slide. So here's what patient experience looks like at Fulcrum. As you can see in this, we have five major components to patient experience. And really what we want to do here at Fulcrum is really talk about advocacy, access, engagement, and policy, and also communications. So we here at Fulcrum want to make sure that we commit our resources, our initiatives, to make sure that we are really partners in advocating for you, and understanding what advocating looks like from your perspective, and how do we do that the right way. Again, access can mean many different things, but also making sure ultimately that you have access to the therapies that we're developing, and anything else that hinders access and resources that you need to make sure that the warrior community is taken care of. Engagement, things just like this town hall is having this dialogue, meeting myself, Dr. Bill, Alex, and Mel, is we really want to make sure that we show up and that we engage with you, and that you just don't know me from Fulcrum, that you feel comfortable engaging with everyone that you see here on this town hall this evening, and feel comfortable in knowing who we are as individuals, to make sure that we have your best interests and be the best partners we can. And also policy. Dr. Billy mentioned as well, some of the town halls really talk about policy and how we can move that needle from a legislative perspective, to make sure that also helps with advocacy and access to medications as well. Next slide. In our mission here as we talked about it, is you see a lot of common themes throughout our presentation and what patient experience focus on. As we talked about building relationships and how do we cultivate those relationships and build that trust and that respect. The partnership again, and also making sure that we support education and access and also advocacy activities. It's not also always about the science, but we know that there are other things in the community that are mentioned here that really will impact the warriors and the caregivers as well, and then make sure that we partner and address them as well. We talked about developing the relationships with other partners that you have in the community, such as the consortium, if there's other organizations, CBOs, even professional organizations and medical organizations that will help us move that needle. We want to make sure that we partner with them as well and represent. And this is really important to us, is making sure that we represent your voice and that's represented in Fulcrum and vice versa. And we truly do mean that we are truly here to be partners. We want you to feel comfortable to put things in the chat. There'll be some more contact information as well at the end, to feel free to reach out to any of us at the team, no matter what our position, what our role is. Mel mentioned that we are small and mighty, and we are a very small company, but everyone is very much passionate about what we're doing here and are very connected and committed to the sickle cell community as well. So next slide. All right, so my commercial break is over, and I think now we get to go into the really great, exciting part of the town hall agenda, which is our Q&A. So I know we've seen a lot of questions in the comments, so I'm going to pause for a second and bring up Dr. Bill, Mel, Alex as well. Sharonda, are you coming up as well? Are you popping in to help me facilitate? Dr. Bailey.
Hello, I'm here.
Great. So we've allotted quite a bit of time for this town hall. This is very important to us. So we want to make sure that we start addressing the questions that were in the comments. So I would just say bear with us a second because there was a lot of great content.
Quite a few.
In there.
First off, that was fantastic. That was a wonderful overview. I really enjoyed those analogies to help visualize what was happening at each space. So thank you very much for that. We have quite a few questions, and those questions go all the way back towards some of the stuff that was occurring at the beginning as we walked through the science. So should we start at the beginning, Danielle?
Yeah, I think there, and correct me if I'm wrong, please give me a little grace, everybody, if I've missed your question or not. We definitely are documenting, so if we don't get them during the Q&A, we will make sure that we follow up with the information through the consortium, or if we're able to reach out to you directly as well. So I want to make sure we have that disclaimer. So I think one of the questions that was asked earlier in your presentation, Dr. Bill, was Derek wanted to know if the dosing in the animal study was the same as the human study. There it is.
Yes, it was. Some of it was much higher, but there was dosing that was equivalent to what's going to be in the human studies.
All right. Well, that was very easy. Thank you. The next question that I see here is from Francine Baker asking, what was the delivery route? I believe she meant the same as that dosing, the delivery route in which they're dosed.
It's once a day oral. Does that answer the question?
I think we'll have to get that one from Francine. The questions were coming through as we went.
Yes. And then I think the next question we have here is, there we are.
How's the drug different from a hydroxyurea, which also seeks to increase fetal hemoglobin? Yeah, a hydroxyurea seeks to increase fetal hemoglobin in a different method. And it does it by actually going to the bone marrow and causing myelosuppression, which then turns on the fetal hemoglobin. Ours does not do that. Ours goes directly to the DNA source, which turns on the fetal hemoglobin.
Now, several of these questions, I believe you might have answered. Well, you did answer as you went. For example, does hydroxyurea complicate the process of how your medicine works? And you talked about why you cannot be on hydroxyurea to be in the trial.
can I be a little more specific with that? It does not affect how the drug works. It's a safety issue, but the two drugs don't interact with each other. And for that first part of the clinical trial, we actually had a lot of patients that were on hydroxyurea, and we showed it still worked even with patients that were on hydroxyurea.
All right. I think there's another
I think
question about just want to make sure we address this is why have you chosen not to have a placebo during this trial? So Bill, I think Dr. Bill, you can clarify that one.
Well, the placebo trials are usually the phase 3 trials, as I mentioned before. Those are the ones in the later stage of development where we're looking to see how effective the drug is, and that's when the placebo comes in. For these earlier trials for safety, a placebo is not needed.
Fantastic. Okay. I think it has, a lot to do with the phase that you're on. I had skipped that to come back to it, but we got that one now because the next few questions were all sort of similar to hydroxyurea. Is hydroxyurea the only drug warriors will have to discontinue? And there were a number of different questions here about, well, what other medications do they have to stop taking? Can they be on other treatments and other modality treatments and drugs during this trial? You did go into a lot of that, but will you just capture that again?
Sure, absolutely. So the 4 drugs we mentioned, right? There's hydroxyurea, which they can't use for the clinical trial. We currently are also not allowing the other 3 drugs that were mentioned, voxelotor or Oxbryta, crizanlizumab, which is Advetco, as well as L-glutamine, which is Indari. So we can't be on any of those for the trial.
And so I think that covers quite a few of those questions, Danielle.
Yes, I would agree.
And that skips right on down to Velvet Brown, wondering about comorbidities. So I believe her question is the number of sickle cell warriors. We live with quite a few other comorbidities, and whether or not you all have any information or data on how this might affect those.
I think we would have to look into the specific comorbidity itself. What I can say is that with our product, it will improve sickle cell disease. But if there's other end organ, unfortunately, that end organ damage has already happened, and so it probably will not help there.
I think the top 3 comorbidities that we see a lot are lupus, diabetes, and moya moya. Moya moya is the one that's not often spoken of, but that might be some of what is being referenced in that question.
Yeah, and our procedure will only, as far as we know to date, we're still early on in our studies, but it will probably only affect the sickle cell disease.
Francine mentioned that what you talked about were very severe criteria, that this seems very restrictive. Why?
This is to make sure that there is an appropriate risk-benefit balance. So patients who are severe and are taking this probably have other risks that outweigh that of the small risk we have for cancer. So taking this drug would be beneficial to them above that.
Okay. What's the next one? Danielle, I'm looking at.
What do you have of Dr. Bailey? I saw a question, some safety questions about the risk of cancer. So one question, there it is from Derek.
Would the risk for cancer increase if you're on Pociredir longer? It was said phase 1 would be 12 weeks. Is that long enough to measure the risk? That for that first part of the question, being that we're early on the study, we do not have answers for that. That's going to take longer for us to establish. Phase 1 would be 12 weeks. Is that long enough to measure the risk of cancer? Usually phase 1s, because they're so short, and this is with any phase 1 trial, to measure cancer in that short amount of time that's not long enough to do that. But that's with any trial in general, not just this one.
Yes. There's another question, and actually, I'm going to punt this to Bill and Mel because it talks about kind of access and coverage. Bev asked about the eligibility for assistance for Procedure.
Mel, you're on mute.
Mel, you're on mute.
Assistance, is she referring to just being a part of the trial, and how can we make that process
About
easier?
insurance, I'm sorry for the clarification. So she stated that insurance isn't always is always a concern with the community and clients that are not eligible for Medicaid or Medicare. So those that do not have insurance, would they be eligible for assistance?
Well, at this stage, we're still in the clinical development program, so obviously we will ensure that a patient who's a part of the trial will have full access to the therapy during the clinical trial phase. Obviously, once you go commercial, depending on your insurance carrier, that needs to be negotiated. But at this point in time, where we are in phase 1B, any patient that's eligible for the trial will definitely have access to the therapy, but also other resources that we're making available to them as a part of the Pioneer study.
Thank you, Mel.
Mhm.
And then I see Francine has another question for clarification about improving quality of life, and that this is not a cure. So is that correct?
I think it's too early we're too early in the clinical process to speak about the quality of life or cure.
Yeah, for us right now, we see it as the disease-modifying therapy. That's where we are. As Dr. Bill mentioned earlier, we're seeing some very impressive levels of HbF induction, which gets you to that level of curative. But right now, we see this mainly as a disease-modifying therapy, which has tremendous potential to increase fetal hemoglobin, which in and of itself has a profound effect on the symptomatology of the disease. So that's our focus right now on continuing to cultivate as disease-modifying therapy.
Yeah, and I think that, Francine, that's exactly it. In the past, the consortium, we have sort of broken up our education into disease modifying and what we're calling curative.
Mhm.
And this would fall in the disease modifying and not curative.
Yep.
And I see it's here, so I'm really glad. The question that's also on YouTube, but it looks like we'll get there. I was looking that up. So do you want to take the next one, Danielle?
Absolutely. So if I'm going out of order, please forgive me, everyone. But I do see a question about being off of
260
being
days.
off of it for 260 days. So I just want to make sure that we clarify that question and that criteria.
It's 60 days.
60 days.
60
Yeah,
days.
not 260, 60.
Yeah,
Okay.
60.
And we have another question from Pat. Hi, Pat. If you're on there. She has a question about if you have renal disease, can you participate in our Pioneer trial?
Yes, you can. In fact, that would be an inclusion criteria for this trial. What you cannot have, I just want to clarify this, so you can't have renal failure. So if you're on dialysis or require a kidney transplant, that would exclude you from the study. But if you do have renal disease, you can. That would be an inclusion criteria for the study that would allow you into it.
All right. So I think the next question is from Emmett. There we are.
Mm-hmm.
I know Dr. Billy would love this question.
He loves this question.
She's got her doctor hat on. I know.
I think I've asked this question.
You have.
This one is coming to us from our YouTube audience.
Yes.
Okay, what is the mechanism of downregulation of BCL11A, and how similar to XSL is 6-fig in terms of outcomes? This is a great question. We are currently looking into this question, but cannot provide the answers to date. But we have asked this question internally and are doing studies to see how we can answer this question.
Good answer. I love it when someone says we don't know, but working on it.
Yes. All All right, what do we have next? So I think April, you asked the name of the drug. So I will actually let Mel take this one because there is a method to the madness and a process that I think he can explain better than anybody.
Should we spell it for him too?
Yeah, pociredir is how you pronounce it, pociredir. And we went through a pretty rigorous process with the FDA and other healthcare bodies globally to come up with that name. But the FDA is very specific on the stem and also the prefix and what you can use. And they felt that was a name that fit the mechanism and the asset in the appropriate way. But the name is pronounced pociredir. So we actually had a competition internally. I think Alex won. Pociredir was the the best way, is the way to pronounce it. Yep.
There are a lot of rules that go to how these things must be named.
A lot of rules. Yes, it's a rigorous process. We actually submitted 5 or 6 names to the FDA, and they said, no, we're gonna give you a name. So they took out the ones that we wanted and put a little change to it, but it was still fine. We thought it was appropriate. It made sense, and we like it.
All right, I think our next question is from Travis.
Yes, good question. It's for Bill.
Okay, how long does this medication take to be therapeutic? And do you need frequent lab works, like when you start Hydrea? We have seeing that fetal hemoglobin increases almost immediately. It takes about 2 weeks, though, before we start to see some real clinical increases in fetal hemoglobin. Now, do we need to do frequent lab work? Well, being that this is a phase 1 clinical trial, there will be lab work. About every 2 weeks, we will need to draw labs for this clinical trial.
Now, I would like for us to grab Derek Robinson's comment. He said that I asked about the length of the study because I think it was mentioned that folks who were on the therapy that has the same mechanism of action as pociredir were on that therapy for a long time. I thought the implication was that it was the length of time which led to cancer in that therapy.
There is certainly an element of length of time, and I'm assuming you're speaking of tazemetostat. There's certainly that element. That the length of time may have contributed to that. As we said, sometimes we see many things that may contribute to it, but we can't pinpoint the exact one thing because other things may have contributed to the cancer in that study for tazemetostat as well. And that includes the other therapies that the patients were on. So hope that answers your question.
Okay.
We have, I'm gonna bundle the next 2 questions because I think they're referring to inclusion exclusion criteria. As far as what's the age criteria from Caroline, and then Pat asked about pulmonary hypertension, if you could participate.
Yeah, there is an age criteria. It's adult patients, ages 18 to 65. And then, I'm sorry, pulmonary hypertension was the next one?
Yeah.
Can you participate? Yes, you absolutely can participate. In fact, that is one of our inclusion criteria. If you have pulmonary hypertension, that would get you into the study.
I think I am seeing all of them that are here.
I think you're seeing.
If you all have any additional questions. Anyone in any of the platforms, your questions are appearing here, so you can go ahead and type that in. Okay,
got some more.
I just saw another question there.
There we are.
Yep.
Yes.
It says, so let's see this one. Should it be clear if Advecvio, Oxbrita, or Andari, I can't get into this study? That's correct. That's it. Currently, we are not, patients cannot be on any other sickle cell agents.
So I see Ray's question. So I want
patient assistance.
Yeah, is this for the drug itself or the participating in a clinical trial? And I think we can probably answer both as well.
We will certainly have patient assistance for the clinical trial. Yes.
Yeah, I'll just say a few more words. Our goal with our clinical trial is to make this as unburdensome as possible for the community. So we're going through significant lengths to work with our clinical operations team, our vendors, just to ensure that there's really no burden at all additional to being in a trial going forward. So that's something we really are serious about. We listen, as Alex mentioned earlier, we're listening to the community and what they need. It's a big part of what we're trying to do now to ensure that it's as easy as possible to participate.
All right. There we are.
We'll be going on Oxbryta. Is it safe? What's the cons? And does it elevate fetal hemoglobin levels? I'm in Florida. How can I get on pociredir? With Oxbryta, what I suggest is that they have labels, which are called USPIs, United States Package for Oxbryta. And that will list out all of the side effects for it. Oxbryta does not elevate fetal hemoglobin. What it does is that it stabilizes red blood cells and hemoglobin. It says, I'm in Florida. How can I get on pociredir? We will have a site in Florida.
Yeah, University of Miami.
Yeah, that's currently we're trying to get that up and running.
Mm-hmm.
So you will be able to get on it there, and we can provide more information for you on how to contact that site.
All right. Derek has a follow-up question next about our phase 2/3.
If the trial moves to phase 2/3, does the inclusion criteria change? Yes. Yes. Doing this phase 1 trial will help to help to create the inclusion criteria for the phase 2, and then the phase 2 will help to create the inclusion criteria for the phase 3. We'll use all of that data moving forward as we end up designing these clinical trials.
This is great. We're getting a lot of follow-up questions, so this is very exciting. So I think Emmett has a follow-up question as well.
As I understand, is there an increased risk for leukemia with sickle cell? Does the cancer risk as assessed thus far appear to be in excess of that baseline? We can't answer that question. We are not far enough along in the study to be able to answer that question.
All right. Nice. Here we are.
Okay, clarifying. And would it be 60 days to be off of all other sickle cell therapies, not just hydroxyurea? That's correct. That's correct.
Yeah.
Now, Pat Corley, welcome. Well, Pat Corley is asking if you would list through your identified sites where your trials will be.
Yeah, I can. You go into it, Bill?
Yeah, I mean, I can look those up. Do you have those?
Yeah, we have right now we have 2 sites that are fully active. University of Augusta in Georgia. We have also in Dr. Rivlin's site in the Bronx, New York, as well. And as Bill mentioned, in the next 2 weeks or so, we should have a University of Miami. So those are the 3 sites we have planned now, and hopefully between now and the end of the year, we'll have an additional 4 or 5 sites as well. Where we're looking at the East, a lot of sites on the East Coast, in particular, New York, the Carolinas, Florida, Georgia, I think even Mississippi. So we are trying to expand our overall reach across many different states within the United States. So,
and I think, and I think Ray has a question that I think would tie into that about the sites, about fairly qualified health centers.
Will you utilizing any FQHCs for the study sites, or only hospital-based clinical and institutional settings? I'm going to have to ask a question. What are FQHCs?
They are really qualified healthcare centers. So,
we may be using those. We may be using those.
And I think I can go ahead and give you a follow-up that I guarantee will come from our international group. Are you coming to them in any capacity? Where?
Yes, we are looking to go outside the U.S. Right now, we are looking to go into the U.K. as well as into South Africa.
Yes. Mm-hmm. Yep.
Yeah. I definitely see some shout outs to states and cities that you'd like for us to go to. Well, we'd be happy if there are any specific centers that you know of or physicians that that would be super helpful for us to make sure that we try and reach out and have access and sites in those cities. So if you know anybody, we connect us or drop a center, drop a name in the chat for us. Thank you.
Especially Texas. I'm a Texan, so I see a bunch of Houston shout outs here. So let me know. I'm more than happy to do what I can to move the needle in the great state of Texas. So
There we are.
There we are.
There we go. How do you gather information from patients regarding their symptoms while undergoing the new treatment? Additionally, do you recommend or support participants in maintaining journals to document their experiences? Yes, we do gather that information. They have every 2 weeks. Patients will need to go in to see the doctor and describe to them any symptoms that they had. They will also get regular follow-up calls where the clinical coordinators will call to ask them how they're doing. Do we recommend patients maintaining journals? Sure. I'm not going to say no. You know, it certainly would help the trial, but it's not required.
Well, we have Derek that's saying, don't forget Jamaica. Please don't forget Jamaica. So please know that.
Thank you for joining us, Sharonda. You were missed.
Hey.
Unfortunately, Jamaica, unfortunately, we looked in Jamaica. We'll not be going down there for the study.
All right. Who else do we have in here? They are shouting out all of the states, and we are going to capture it.
Absolutely.
yes,
Yeah.
yes.
They are really shutting it up over here. Let's see, what else do we have down here? Any other questions? Now, this session is the Q&A for you guys. We built this in the way we have just because we wanted to give time and space for any of your questions. So I do see all the shout outs. Yes, we will let them know where they need to go down. I mean, Texas is, listen, Mel, you got to make sure you handle Texas.
Exactly. Dallas, Houston, Austin, San Antonio, all my old stepping grounds.
I see Canada as well. Someone asked for Canada.
Yeah. We are not planning on going to Canada at this point.
Not yet, y'all. Not yet. Oh, we got a question coming in.
We do have a question. All right.
Let's see. My question may not apply in this setting. Have there been any discussions or research into therapies targeting the sickle gene in individuals with sickle cell trait? In other words, editing or correcting that gene so that individuals won't pass it to their children. I don't. That's a question I'm not sure I can answer. We can answer. I don't know of any at this point for sickle cell trait. But you know, that's something that we would have to look for in the literature. But I don't know of any at this point.
But it is a good question. We do do a lot of stuff with educating for sickle cell trait here at the consortium, especially with a lot of our events. So we will keep that in the back of our minds to ensure that when we're talking to industry, that this is a concern for the community. So thank you for that question. The next question is coming from Ms. Pat Corley.
Let's see, I'm sorry. What clinical or labs specifics would be needed to BA sites? Can I get some clarification on that question?
I believe it needed to be a site. Yeah.
Oh, okay. Specific. We're looking for standard CBC, standard chemistries. The one particular lab that it's going to be sent out will be for the F cell measurements.
And I hope that answers your question, Ms. Pat. And if you need a clarifying question, please go ahed and put that in the chat as well, and we'll make sure that gets answered.
I will say we are currently not using a central lab. Although, with the exception of a few labs, we are using the local labs for those measurements.
Okay, good to know.
Yeah, Sharonda, if we don't answer a question fully and you get a follow-up, let us know, and we'll get back to them immediately through our medical affairs team and or Dr. Bill.
Okay, great. Thank you. I love that. Thank you, Mel. And you know, I'll be emailing Danielle, pounding her like, these are my questions.
No problem. Email me, text me. You know how to get to me, Sharonda.
I do. So I don't see any other questions coming through. I do see a lot of places where they want you guys to come, so that is great. Yes, we need everyone come to. They said South Carolina. Okay, so Pat said, "Can doctors?" Let's go down to see what Pat said.
Mhm.
And she's saying, yeah, there we go. Can doctor offices be sites? Oh,
Um, as far instead of hospitals, can we get more clarification? Yeah, yes, a private clinic can be a site if they have the appropriate
Infrastructure.
infrastructure. Thank you. They have the appropriate infrastructure there to set it up. They certainly can.
But also, too, if there is a site nearby and let's say you don't participate, you don't actually visit that site, your physician can always refer you to a particular site as well to be a part of the trial. So there are many mechanisms. If the trial is nearby, there are many mechanisms by which you can get yourself involved as well.
well, can you go back just real quick about what Pat is just asking, though? So you're saying that the doctor's office can be a site if they have the infrastructure, but how do they, do you just refer? Like, do we just tell the doctors, hey, this is the thing that's going on? And then they contact you to ensure that they have the infrastructure, or how does that work?
Yeah, they would contact us. They'd contact me. If they're interested in being a clinical site, we would have to go through the process of making sure that they have the infrastructure.
Okay.
And going over the details of the clinical trial to make sure that they can that they want to participate, as well as I mentioned before, make sure they have the appropriate patient population.
Understood. But the contact for Fulcrum would be you, or would it be
myself, or it could be anybody. Myself would probably be the best one to contact.
Okay.
Mel is me. Mel's well.
Got you. So we'll make sure, Pat, to give you that information so that if there is a site that you think would be interested or be good, because you know this community very well, that you think would be good, then I will make sure that you get that information so that you can pass it on. Okay, so we do have another question. Joy, if you can put up Alyssa Kofri. Apps, girl, you got me. There we go.
Proximity to the site to be in the study. How often are labs? I think what we're asking is how far away do they have to be in order to be in the study? We certainly have not set up any distance to date, right? And we will provide transportation into sites if they need it.
And I think that's where that question is coming from, because providing access to care also means that sometimes we just don't have the transportation.
Exactly.
You have so many times that you have to go get labs, and especially if you got to go back and forth, what does that look like for, the warriors and the caregivers? So you're saying that you will provide transportation?
Yes.
Okay.
Absolutely. Yeah. Absolutely. How often are labs? The labs will be taking place about every 2 weeks. Every time you go into the clinic to see the doctor for the study, it will be about every 2 weeks. It should be a single stick, with the exception of the first and the last dose. In the first and the last dose, I think there are about 5 sticks that will occur, unfortunately.
Good to know. Caroline had one question. There we go. Also, a concern is that there are people with the trait that are symptomatic. Are they excluded?
Unfortunately, trait is excluded at this point.
Yeah. But we're making noise about it, Caroline. So keep talking about it, making sure that you are talking to your centers and making sure you're talking to your doctor about it, so that in the future there may be something, but right now there's not. Let's look at what's happening with Derek Roby. I love Derek. He have all the good questions. Pull up his question. There we go.
Okay, the comment about gene therapy that would stop the gene passing to children is germline gene therapy, which I don't think is allowed. Somatic editing is what is being done now. Doesn't affect the germline. Dr. Bailey may comment. I'm going to pass this one. First question, it's not mine. Dr. Bailey.
I'll pass that one.
Yeah, I think Dr. Bailey had to hop off for a second. She may not comment at this time, but we definitely wanted to thank you for. Oh, there she is.
I am here.
All right.
I'm here, everyone, but my computer is dying on me. So if I stop abruptly, that is why. Pull the question back up for me. I'm sorry. I heard my name.
There it is.
The comment about gene therapy that would stop the gene passing to children is germline gene therapy, which I don't think is allowed. Somatic editing is what is being done now. Yes, that is something I've commented on before. And I do think it's important that we recognize that it's a question that does come up, and it's a realistic question based on, our average understanding of gene and gene therapy. But there's different genes. There's the genes that affect your children and their children. And we're not talking about epigenetic modifications, but like somatic, which is what Derek is referencing here. And then there's the germline, and then there's somatic. So, I mean, there's the different types of genes. I'm getting all over the place. But what we're manipulating right now is not that type of gene. It's affecting you, but not the next generation or the next. And so we use the term curative because of that gene editing. But in those cases, it's curing you. You would still pass sickle cell on to the next generation. Not sure if that's where that question was coming from, but I heard my name, so here I am. And that's not what we're doing in any space right now. No one is doing any kind of research that is going to alter the genetics of the next generation. And there's a lot of debates about the ethics of that and whether or not that will ever be allowed.
Thank you.
I'm seeing a whole lot of questions here about the genetic makeup of the sickle cell, and I can answer most of these in one answer. So for this clinical trial right now, what we are looking for are patients with SS, S beta plus, or S beta 0. Those are the genetic makeups for this clinical trial. Fortunately, we currently are not enrolling SC patients. That may change, but we currently are not.
Can you repeat that for us one more time?
Sure. The genetic makeup for the sickle cell patients are SS, S beta plus, and S beta 0.
Okay. So that would answer the one for Pat that she asked on there. Thank you so much for that question, Pat.
Yeah.
And I think
Elisa Fink as well.
Yeah,
yeah, there's a whole bunch I saw in there about the genetic makeup. Yeah.
Pamela as well.
Ours is such a large family of warriors that we have, SD, S Punjab, SO. There's so many of us that connect now.
Yeah.
So it's really good to specify that.
And Pat, I saw your comment about California. We are looking to go to California.
Yeah.
We would love to get out there.
Yes.
We are working hard to get out there. Yeah.
Yeah, we're definitely trying to make sure that all warriors have access to all of our clinical trials, if not the current one, but for future trials. So all the places everyone shot out in the comments, we are duly noted, and we'd love to be as many places as possible. So continue to do that.
And one of the tips that I'll give the audience is that they've already said that contact them, let them know, ask the questions. So make sure that you are actually doing that. So when we give you guys the information, the contact information or the email that we send out, make sure that you are, talking to them and letting them know that you do have a community that wants this to come your way. And I know, Pat, you gonna make sure. So we would definitely get you that information. There is another question that I thought we probably should read. So the one I'm looking at ASH presentation for 2020. Yeah, let's look at this one. Okay, is there a link somewhere I can read more recent information and data? So they're just asking, do you guys have any more information?
So I've asked those questions before too, you all, and it's just where can we get a little bit more information on the data that you present? Where are your papers published? Is there a place where we can go and read? It sounds like Emmett Major is one of the nerds amongst us. Emmett Major, I mean that in the best way possible, but I am too, and we like to read. Tell us in PubMed where we can go, and if we don't have access to PubMed, how can we get that good data?
Yeah, there's also one other question I'd like to pose to the town hall and the community. I know we asked for any medical centers where you'd like us to participate in a clinical trial, but also we want to support you from a community aspect. So in any of these locations, please give a shout out to your local CBO so that we can make sure that we establish this relationship with them and they get to know us as well. So don't forget to drop a note to them as well, as we'd like to get to know your local partners and advocates in those cities as well.
Yep. Awesome. So I think we can combine the one that I asked about where is the data with Derek's question of will you be presenting at next week, ASH San Diego 2023.
Also, actually, we had an answer to that previous question. One of our Fulcrum employees is on comments there. If you go to the Fulcrum website, you can find many of the archived presentations.
Yeah.
I see it. There's the link. And will you be at ASH next week?
Yes, we will.
It's gonna be Ash.
Yeah.
Awesome.
Nice. All right, so if you guys see them at ASH, pull them aside and get your questions answered. No, don't mock them. Just
It's okay. It's okay. Okay.
Let's see. I don't see any other questions, Dr. Bailey or Danielle. I don't see anything else that people like ask you.
I don't think so. I don't
think
see
we got
anything. Yeah, I think we got
Hey,
it.
Ray says sure will, so she's gonna pull y'all to the side. So just know that
There's our booth. We got a booth number in the chat. So
It'd be awesome.
You
All
can
right.
mob them. You guys can mob them. They can take it. They
they
can mob
can
them.
take it.
Interesting question. Sorry, Pat's got a question here. What about hemoglobin S with high persistence of fetal hemoglobin? Are they candidates? That's an interesting question. Are you asking about patients that have hereditary persistence of fetal hemoglobin, or
Sounds
just
like
those? If that's the case, I'm not sure. They're not excluded for any reason, but I'm not sure why they would want to join.
Yeah, I have a feeling that they wouldn't meet the criteria as far as
Severity.
well, yeah, severity. If you have
Right?
HPS, some do, but if you have a
Yeah.
high persistence of fetal hemoglobin, you might not meet the severity.
Yeah.
And the lucky you. Are there any other questions?
Story. Oh, okay.
Yeah. Thank you for the invite.
Mhm.
Yeah, let's pull that invite up right here. We got people that want to invite you guys over to talk with their CBOs. So yeah, they're shouting out their CBOs. I love it.
Thank you. This is
Yeah,
awesome.
absolutely.
Absolutely.
Yeah. I see another question about beta thal.
Yeah.
Ask a question about beta thal. Is beta thal a different type of disease, or is it stem from sickle cell disease? It's similar, but they're different diseases.
And I think we've done a class on this in Warrior University, and it's probably, you can find it at the Warrior Network, scwarriornetwork.org. Little shameless plug there. Lots and lots of classes and education up there. I taught that. So if it predates Warrior U, then perhaps we can go through and teach that one again about these different and what they are and how they work. But thalassemia is a very different disease. And then you can combine sickle cell and thalassemia in some very interesting ways.
If I can do one plug here, as a follow-up to this town hall meeting, we'll be sending out information via our new website. To go to our new website called pioneerscdstudy.com. So in about a week or so, we'll be launching that, which will provide you with more information about the study. So we're excited about that, pioneerscdstudy.com. So you'll hear more about that from us from the town hall follow-up.
All right. Derek has something else.
For the future, I was thinking of patients who may not have the severity, but have reasonable levels of HbF, say 9%, and a therapy like pociredir would be good to push them to 20% levels. Yes.
Yes.
That certainly would. Yeah, that certainly would be great. But as you said, I think the key word there is for the future.
It's definitely a very interesting conversation, Derek, about how a lot of times you have to have a certain level of severity to even get into many of these trials.
Yeah.
We don't want to get to that point. So what about before you get there, but you're trying to stop it? And that is definitely a conversation worth having.
Yeah, absolutely. Some of the end organ damage, patients may not have pain crises, and they just may come up, right? Some of the work. And, the whole idea is preventative versus, reactive here. And we'd rather be preventative instead of coming back and trying to, fix something that's already happened.
I think also when you're doing quite well, we forget that this is a progressive disease.
Yeah.
And you may not always be doing quite well.
Yeah.
And it's easy to forget the damage that's being silently done.
Yeah.
I don't
So
understand this question.
can
Oh,
our,
yes, I do.
can reports be used for required labs is what I see.
Yes.
Maybe, yeah, maybe. I think they, I'm going to leave that up to the site and the doctors to let to answer that.
Mm-mm. I read that as can our parts be used? I was a little lost. All right. It looks like we're really winding down on the questions.
Yeah.
But you all, if you think of others, you can continue to reach out
Yep.
to ask those questions, to reach out to Fulcrum directly, reach out to us to allow us to share those on your behalf. And we will do that. And in some capacity, provide the updates on those questions. And we might just have to bring you all back. I think we're planning to do that anyway.
We are. You guys can't get rid of us that easily. We are definitely planning a part 2, or as I would say, a second date in January. So after the holidays, we hope to see all of you there, and tell a friend as well. So yeah, thank you for dropping that nugget as well, Dr. Bailey.
Absolutely.
And I've put my email in the chat, and I believe one of the Fulcrum team, they will give us the contact information. So as the follow-up email to all of you that registered, there will be email that will go out giving you their contact information. But if you have questions and you want to contact us at the consortium, you can email me at sykes@sicklecellconsortium.org, or you will have that email sent. Yep, and Danielle just put her information in the chat as well. That's our Danielle.
Yeah.
She put it in the chat as well. And so we will make sure that you have the contact information. And I do not see many questions. Now I see lots of thank yous, hand claps. Oh, I don't have my little hand clap sound on tonight, but that's okay. If you were on my live yesterday, you know I overused it, but I love celebrating the people. So I thank everyone for being here. I don't see any more questions in the chat. Derek is saying thank you. Everyone is saying thank you. It's a thank you. You get a thank you. You get a thank you. We all get a thank you. This has been a great town hall, and I know we're going to do a part 2 in January. So be on the lookout for that information. Share this information. Now, this is a live on Facebook and all the things, and on YouTube. So you can go back and listen to it and then ask questions from there. Like whatever you need to do to get the information. Because for me, I got to listen to it multiple times to get it all. Because this was a lot of information. So make sure that you share it with your communities, because we do want to hear your thoughts. We care about what you have to say and what your thoughts are. So I do want to hit it back to Dr. Bailey and Danielle so that they can say their final words for tonight. So you guys can go back and give you back to your families, go have dinner and all the things. But thank you guys for coming out. Thank you for registering. And thank you for being here for our final town hall. So I'm going to give it back to my lovely Dr. Bailey, and then she can say her final words, and then Fulcrum can say their final words.
All right, I'm going to go quickly. My computer is dying. It's going to shut off any minute. So thank you. I could not find an extra charger, and I'm not at home. So thank you so much, Fulcrum, for joining us. Thank you, everyone, for coming to this town hall, this final town hall of the year. We are kicking it back with another town hall with Fulcrum, and we will be sharing that information. And your questions in the meantime, you can definitely ask those anytime you want. We are committed to providing as much information as we can, and there will be more town halls from Fulcrum and many others that are scheduled for 2024. Partners, don't forget, we have a partner meeting tomorrow, our partner meeting of the year, to let you all know that credentialing is open. And so if you are interested in partnering with the consortium, you have until January 15th to credential to ensure that you are a partner in time for the March Leadership Summit, but credentialing is rolling. So ask your questions, learn more about it. You can credential after January 15th, but you must to attend the Leadership Summit. Thank you again so much, Fulcrum, and all of you for joining us for this incredibly important information and for answering all of the questions. I think it's incredibly important to be able to, what I like to call, gracefully coming to the extent of your knowledge, which you have done wonderfully by saying, I don't know, but we'll get back to you, and for just answering all these questions. So thank you very, very much for that. Danielle.
All right. So I couldn't have closed better than Dr. Bailey, but I can honestly say I can speak for myself, Dr. Bill, Mel, and Alex, that we are truly humbled and that everyone spent, I think we're at 1 hour, 53 minutes and 30 seconds on a Thursday, that you spent your afternoon and evening with us to learn about us and also give us a lot of information about what you want to know and start that dialogue. So we are truly humbled and thank you for spending this time with us. Like Dr. Bailey said, we will see you soon. We will have a second date in January, so you can't get rid of us that soon. We put our emails in the chat and we truly, truly mean it. Please reach out to any of us on the team. We really do want to hear from you. We really want you to know who we are as people, as a company, and our commitment, and vice versa. So without that, I'm going to say thank you again, and I am done. So thank you.
Awesome. That Alex or Mel, do you guys have anything, or we're closing up?
No, we're good. They yell
I'm
fantastic.
good.
All right.
All right. Thank you guys so much for joining us. We appreciate it. And now, everyone, we are closing out of this town hall. Thank you so much for joining us again. We appreciate everything. We appreciate this community for showing up. When we call, you guys show up, and that is just amazing. So I don't have to say anything else. We're done for the night. Go having a great time with your family. And yeah, make sure that you are keeping in contact with us because we got a lot of things happening in 2024. If you saw the newsletter today, I gave y'all little updates on the dates for our events for next year because I need y'all to start planning now. The Warriors Convention, the Leadership Summit, the Caregiver Summit, it is all coming back in 2024, and we want to see you guys there. So that's it for us. Thank you so much again, and have a great night. Bye-bye.
Good night, everyone. Bye.